Good morning, everybody. Welcome to Science Saturday. It is a beautiful day. I am down at the University of the Virgin Islands Center for Marine Environmental Sciences, and I am so excited because today we are talking about mangroves. And you guys know that I love mangroves, and we've done a Mangrove Science Saturday before, but this month, we are looking specifically at baby mangroves, and this is really exciting. This is kind of similar to last month. For those of you who are with me, we are on St. Croix at the Nature Conservancy Coral Innovation Hub, and we were looking at how they are microfragging and growing corals in order to help with the coral population. Today, we're doing something really similar, only something totally different, because we're talking about baby mangroves. So, I am joined today by a fantastic team of researchers at the university, and we're flipping the camera around, and who are we going to start with? We are starting with Dr. Kristen Wilson-Grimes. Good morning.
Good morning, everyone. Thanks for joining in. My name is Dr. Kristen Wilson-Grimes. I'm a research assistant professor here at UVI, and I am leading this restoration project. And I have some friends with me today, some amazing women scientists who are going to introduce themselves now.
Hi, good morning, everybody. My name is Allie Durdall. I am a watershed and marine specialist at the University of the Virgin Islands, and I work with Kristen in our lab. And we've been working really hard the past few months to get this mangrove nursery off the ground, so we're so excited to show you today.
All right, awesome. And
hi, I'm Dr. Heather Stewart, and I'm a new postdoc here on the VI EPSCoR Ridge to Reef project, and I'm here to help restore the mangroves in the UVI.
that sounds super cool. And you guys know our favorite Science Saturday intern, Smush Mason, the amazing. So excited for a fun day, right? Yep. All right, awesome. So, what do we have going on?
All right. First up, we're going to go and take a tour of the mangrove nursery.
Ooh.
And then after we kind of talk about the tour, what we have going on here, we're going to come back and we're going to do two different activities. We're going to show you how we process white mangrove propagules. That's a new science term. So propagules are kind of like seeds. You could think of them like that. But what's so neat about mangroves, one of the adaptations that they have is that the adult trees actually start growing their babies while they're still attached to the trees.
Oh,
What? That's crazy.
right.
And that has a special name for it. That's called propagules. So we'll process those, and then we're going to plant some of these white mangrove seedlings that are ready to go into tree pots. So Mason's going to help us out with that. He's going to help us with our intake. But that's the plan for today. Before we do that, I'm going to turn it over to Allie. Allie, why are mangroves so important?
Oh my goodness. Well, this is an easy question to answer because mangroves are so important for so many different reasons. Mangroves provide protection to our coastlines. They help buffer storm surge when big waves are coming through. But they also protect the ocean because they're really good at catching and trapping sediments that could make their way out into the ocean and smother our seagrasses and coral reefs. Mangroves are important also for the animal life that lives in the Virgin Islands. So mangroves provide shelter and nursery grounds to fish, to lots of different species of birds, to all kinds of cool species of insects. And they're great all around for tourism purposes too. People love to get out and enjoy the mangroves. We have mangrove tourism here on St. Thomas. They also collect and trap carbon, so they are an important player in the global carbon cycle, and in that way they can help mitigate climate change as well.
Well, that's important.
Yeah.
And I know from listening to you guys talk a lot, there's a really fun science word that we can learn today, and that is carbon sequestering. Right, guys?
Yes.
Yeah.
Yeah. So
And that's all I'm going to say about that.
I'm gonna let Kristen talk about that one.
Sure. So carbon sequestration, it's just when the plants take the carbon that's out in the atmosphere and they lock it away in the plant tissue. So the stuff that you can see above ground, so their leaves, the stem, but importantly, the stuff that's below ground in the roots and the rhizomes, so these like root-like structures that go out underground. And what's so cool about mangroves is that they can accumulate those materials in the mud and in the sediment for thousands and thousands of years and lock it away. So they're really good. That's what refers to as the carbon sequestration. That's taking that carbon from the atmosphere, locking it into those plant materials, putting it underground for long periods of time.
All right, awesome.
So before we take the tour though, why do we have a mangrove nursery here at all? Well, for those of you who were here during the 2017 storms, you know that our mangroves, they took a hard hit and they're still recovering. So part of this work that's funded through VI EPSCoR as well as by TEMA and other partners down here is to try to see if we can give the mangroves a head start by creating a nursery and helping with restoring the populations that were there and the forests that were there. So that's why we're building this nursery, and that's what we hope to do over the next several years here. And that's why Heather is here.
Yay!
Is to help with a lot of the restoration projects. Heather, do you want to talk about why restoration is maybe important?
Sure. Well, restoration is very important because as the trees start to die off from the changing of the hydrology, so the water was changed with the different storms, you want those areas to be able to grow new mangroves again. And so some of the mangroves that are still surviving are producing these propagules, and then they'll try to go and replenish the area where the mangroves were before, but sometimes they need a little extra help because there's more predation. So there's crabs that like to eat these little mangroves. They're very tasty. Goats like to eat them as well. So what we're doing is we're giving them a head start, and then that way they'll be able to successfully return, and then we don't have to worry about coastal erosion or storm surges. So with more storms coming in later, they're not going to have the same negative impacts that they would have on people if the mangroves are there.
All right, super cool. And I have one question totally unrelated.
Yeah.
Before we do our tour. Heather, you were introduced as being a postdoc. Can you explain that to everybody? We've got a variety of titles here today.
Okay.
So let's talk about that a little bit because it's so cool.
So postdoc means that I recently finished getting my PhD, so I'm now a doctor. And
What?
yay! But I'm not yet a professor, so I'm kind of in between those two stages, and I'm just focused on doing research. Maybe wait for the plane.
Don't mind the airport, guys. We know the drill.
So I'm really focused on research, I'm focused on conservation, and I'm focused on outreach and education. And so before this, I was working in Panama for 5 years, studying the biodiversity of mangrove islands and which islands support the most life, and what factors about mangroves attract certain animals and scare away others. So that's what I studied, and that's what I'm trying to bring here to help everybody here.
All right, awesome. Very cool.
All right, let's go take the tour.
Yeah. All right, guys, we're going on the tour. Say hey, Smush. Hey. All right, we are going over to the mangrove tables. I'm coming, I'm coming. Woo! So cool.
Yeah. So these are part of our red mangrove nursery. So this right here, we're on a flow-through seawater tank. So there's water that's getting pumped up from the ocean to help keep these red mangrove propagules or seedlings nice and cool. So they're soaking in seawater right now. You'll notice that each of these buckets, you might be asking yourself, why do we have different buckets? Well, each of these is are labeled with a different type, a different site. We're trying to keep track of where these different propagules are coming, and I'll just say that all of this work is permitted by the territory for us to collect these, and also by the national park, although we don't have any national park red mangroves yet. But we do have permits from both of those places to collect these. And you'll see each of these has a different label. There's some that has this. You'll see one has, Compass Point, that's right here on St. Thomas. But you'll see we have a couple. Where's Altoona Lagoon? Altona, you guys all know that.
Hello, St. Croix.
St. Croix, all right. Woo! So you'll see these red mangroves have some pretty cool adaptations. And you can see some of them are already rooting, and that means they're ready to go. We're going to transplant them into some sediment. Allie, do you want to talk about some of the cool adaptations that these red mangrove propagules have? In terms of maybe how they float in the water?
Sure, yeah. So what you're looking at here with the red mangrove propagule that maybe isn't so obvious is that this propagule itself, in its size completely here, is actually the size that it was when it fell off the tree. So it's very different from a normal seed from a regular plant. If you've ever planted seeds in your own garden, you probably know they come in packets from the store. They're very tiny. But red mangroves grow their propagules, their original seeds, to be very big before they fall off the tree. And this gives them a huge head start, right? So mangroves are a cool species because they are some of the only species in the world that can grow in salt and in fresh water. But to do that, that means they have to be pretty hardy, right? So it's kind of stressful for them to grow in salt water. So they can take like any leg up that they can get, and that can help them grow in these conditions that are a little harsher. So they grow these big propagules which will fall and float into the water. And another cool thing about them is that when these propagules fall off of the tree initially, they actually float. So if you think about how mangroves are located along the coastline, these seeds need a way to scatter about so that the trees can reproduce and expand. And so by having propagules that float, once these fall off the parent tree, they can move around to other locations where hopefully they will settle in some sediment and grow there as well.
So that's really interesting, thinking about how they're different than anything else. I'm kind of like stuck with this image in my head, going to Home Depot and finding like a bag of propagules in the seed section. That would not be permitted, by the way. But yeah, they're so different.
So check it out. This is kind of how these plants do this. So Mason, check this out. See how they float? And one of the things that's so cool about red mangroves is you can see they're almost floating kind of perpendicular in the water. And that's because they're actually weighted at this end. And that's an adaptation that red mangrove propagules have, is so that they're floating along, they're floating around, and hey, they hit some mud, and then they're like ready to go to start rooting in life position. And then this end of the plant, Mason, hand me one of those that has leaves. This end of the plant, when it's ready to root, it'll actually start then putting in those leaves and starting to grow into the big plant.
Yeah, so when they're on the tree, you'll notice they have this little brown thing here, this little cap. And a lot of ours here have already lost it, and that's because they've been off of the tree longer than this one. And so as Kristen and Allie said, they can float vertically like this, and they have a pointy little tip that gives them a start that they can root into the sediment. But red mangroves are really awesome. There's differences between all the mangrove species, but red mangroves can float for the longest of all these different ones, and they can go several months without starting. And so when they want to float for long periods of time, they'll actually float horizontally so that they can travel through the ocean waters faster. And then when they sense that the conditions are right for them, then they'll start changing their density so that they'll be vertical, and then they will start to sink. Now they can still change their density and go back up, and they can also start rooting like this one here. And some mangroves are known in places as walking trees because the roots can help pull them in one way or the other. And so maybe they have a lot of shade growing by them and they want a little bit more sunlight, so they'll use their roots to kind of pull the tree over one way or the other.
That's so cool.
Yeah.
All right.
Now I'm also thinking about walking mangroves. You guys are putting so many images in my head this morning.
All right, let's turn around and look at the tank behind us. Ooh, cool. So now you can see these mangrove propagules, they're a little bit older. So these were ones we collected back in April. And so you can see they're starting to sprout their leaves. They have a couple of pairs of leaves. Some of them even are about to get a second set. So these are a little bit older. We're keeping them in these trees because we've collected kind of individual information about all of the different propagules that you see in here.
Right, you don't want them to walk away.
We don't want them to walk away, that's right. So we have them kind of contained in here. And these mangroves are a little bit older. So these are some of the ones that we use. These are still red mangroves, but these are some of the ones that we use for events just like this so that people can see them, they can touch them, and they're used for education. And these ones were mangroves that were collected just out here, right behind the university, by our partners at the Virgin Islands Marine Advisory Service and VI EPSCoR. And you can see these ones are probably, I think they're about a year or so old. So they're a little bit older than the ones that you see over here.
Now, I'm noticing one thing. In our last Science Saturday about mangroves, we learned about, Mason, do you remember the name for the leaves that were taking all of the salt? They were the sacrificial leaves?
Yeah.
Am I right? The sacrificial leaves. So these guys who are a year old, I'm already noticing some of these yellow leaves. And so that means that pretty early in life, they're going through that process.
So a really interesting thing about the different mangrove species is that each one has their own adaptation. So the red mangrove, their specialty is that they grow really well in water. So they're a lot of the ones that start islands out of nowhere. Now the black mangrove, which we'll see in a little bit here, they do the best with salinity. So they can actually live in areas 3 times the level of salt in the ocean. And so they are able to live in salt water through salt secretion, so getting rid of the salt like you were talking, and also salt exclusion. So they have their own, it's much different than other trees. They have their own system within the tree that creates almost a reverse osmosis. So it takes the salt water into the tree and it puts the salt in the layers of the tree itself so that it can absorb the water, the fresh water out of that. So they can get like 97% fresh water out of salt water, and then the remaining amount they put through the leaves. Now, different species are more predominantly secretors or excluders. And so the black mangrove is really well known as being an excretor. So if you ever like lick the bottom of the leaf, they'll taste very, very salty.
Well, now I'm gonna lick a mangrove leaf. I mean, that was gonna happen.
But another thing that they do to help, because just like plants in the desert, they don't want to lose that fresh water that they work so hard to get. So they can actually rotate the leaves. So you see right now they're pointing upwards. And so what they'll do when they want more sun to help with photosynthesis and get energy is they'll flatten so that they can get more sunlight. Now when they have enough, then they'll start to close or rotate the leaves so they get less sun exposure, and then that way they lose less of the fresh water from the direct sun on them.
Okay, very cool. That's super interesting.
Kitty, do you want to see the rest of our nursery? We can show you what we're doing with the white mangroves.
Yes, we want to see the entire nursery.
Okay, awesome. So, we're still kind of learning as we're going, but this is the system we have in place right now. When we collect our mangrove propagules, we bring them into the nursery here. We saw already where the reds go, into this big tank over here where they were in their orange buckets. But when we have white and black mangroves, they go into this table right here. This is our intake table. And we soak these guys in salt water until we see that they start to sprout. So what you can see in here are these labeled cups. So again, we're trying to keep track of where they came from. These ones came from Vessup, so right over by the ferry, car ferry dock if you're going from St. Thomas to St. John. And you can see if you look inside the cups that our propagules look a little different. So this one contains black mangroves. They're kind of this medium rounded size. And again, these are propagules, so they germinate on the parent plant, so they start to grow on the parent.
So these are still propagules.
They are,
now,
yes.
So then, is a propagule a seed, just a very specialized seed, or is it its own thing?
Yeah, it's a seed that is germinated on the plant, so it starts to produce and grow on the parent plant itself before it drops into the ground. Wow. Whereas typical other seeds on the parent plant will fall into the ground before they actually germinate and start to grow. Okay. So it gives kind of a little bit of a head start. So you can see we have this cup with the black mangroves, And then these ones are the white mangroves, so they're a little bit smaller. But all of them are propagules, so they've already started to grow. And what we do with these is we soak them, and every once in a while we'll, I guess as often as we can, we come in and we check to see if any have sprouted. So Mason, do you want to help me? Do you want to look at these? Do you see any sprouts in there? Yes.
Ooh,
Yeah.
A lot of them have a little bit.
Yeah, see the green?
Yeah.
So what we'll do with those after that is we process them. So we're going to do that in just a little bit. We're collecting some information about those individual propagules.
Because
and
science
then,
is cool.
It is so cool. And then we will take them and plant them. So Kristen could show you that.
Yeah, so these are our little white mangroves. So each of them has a unique ID. So we know that the information when we collect in the intake, that it's linked to this little tiny propagule. And if I turn this out, you can see that's the little rooting tip. And in
It's so cute.
I know, and in a little bit we'll show you what this turns into and what happens next on the next part of the tour. But we do also the same with those little lima bean propagules, or those lima bean shaped propagules that were the black mangroves, and you can see that then they start sprouting into little plants, and we have some that are a little bit further along, and some even further along, and we'll show you some of those plants at our next table after we take a little walk.
And you can see these leaves are very soft and tender. So these are the primary leaves, which are the ones that a lot of herbivores, so anything that eats vegetation, they really like these soft ones. And as they start to develop, they'll get harder and they'll be more tough to eat and digest. And then it protects the animals a little bit more. So this is the stage we have to be most careful of crabs and caterpillars or goats or anything that might think this is a delicious little treat.
So we're protecting these guys right now.
Yes,
In the nursery.
exactly.
All right, so we're going back over to the table.
Yeah, we're going to show you some of our tables on the other side that have some of the trees that are a little bit older.
Ooh, more tables.
So we just
They're so cute.
I know. So we just saw the white mangroves when we plant them, and then it's really fun. They kind of perk up out of the little peat pellet or the sediment that they're in. We call these their little hats, but you can see, and then eventually they'll bust that open, kind of like what's happening here, and then they grow into these larger plants. And when they're about this size or so, we transfer them into some bigger pots that are called cheap pots that we'll show you over here.
I have to confess, looking at these, and I can't even get like lettuce to sprout. This is why I hold the camera and I'm not in charge of the science.
Yeah, so you can see these are these tree pots. They're kind of specially designed, so they're a little bit larger at the top than the bottom. Gives a lot more sediment for that plant to grow in, to thrive. And you can see some of these plants are doing really well, like some of these. And then, womp, 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 this one, not so well. So there's a couple of things that could have happened. We had a caterpillar in here. That was one of the things that Dr. Stewart was talking about. So every day we come in here, we water these plants, we check to see how they're doing, we check on the undersides, are there any caterpillars or other things? We do a little check on the ground, on the sediment here to see, is there anything that's happening?
Now, what is the soil makeup? You say sediment, but I imagine it's probably not just commercial soil from the garden center.
Actually, that's what we're using right now.
What? I was wrong. I love being wrong.
But one of the things that we know is that we're going to try to experiment. We're actually going to do some experiments with what we grow them in. We know from other scientists who have done this in other places that when you go to put these plants back out into the environment, that they'll do a lot better if you can match what they're growing in here with what they actually grow in out there. So we're going to try to figure out what the best combination of those things are. But right now, we're just trying to see if we can grow the plants themselves. But check out, Allie's going to show you some of the black mangroves that are a little bit bigger over there.
Yeah, so you can see here are some of the black mangroves that have been growing and thriving for a little while. We're really excited to see they're starting to put out extra sets of leaves. But while these plants are little, they are pretty sensitive. So it's a lot of work for us. We got to come in and we got to make sure they're getting the water that they need. They're not getting too much sunlight, which is why we have these fancy shades that we built.
Mason, help me pick this up. We'll get this out. So normally, this thing's on there. And Allie, can you explain why we have this on there?
Yes. So if you think about a mangrove forest on the shoreline, you can imagine there are adult trees overhead, right? And where most of these propagules will drop onto the ground are into places that are shaded by the adult trees. So these trees, when they are little, are actually accustomed to and grow best in areas that offer a little bit of shade, because that's what a normal healthy system would typically have. And so we are trying to kind of emulate that here in our system with these shades that we've built here.
So look at how much shadier it is now with the shade cloth on there, right?
Right, yeah.
Yeah.
So it's not like totally darked out. Darked out? That doesn't feel like the right word. They still have light coming through, but it's like they're underneath the
Canopy.
yeah.
Yeah.
I was gonna say mommy and daddy mangroves.
All right. What is this made out of? So this is just shade cloth. You can actually buy it just at Home Depot. That's where we get ours. We actually double up the layers, so you can kind of see this on the side. We double this up so that we have extra protection. And at some point, we're gonna have to get these plants ready to go back out, which means that we're gonna increase the light. So we'll probably remove one of these layers and kind of get them ready before we go to outplant them.
Very cool.
Yeah.
And here we're lucky enough that we have places that you can go buy products like this. But in other regions where there maybe might not be the ability to go out and buy such products, people will use palm leaves or other types of plant material to create natural shade. And then you just slowly reduce the light, or reduce the shade and increase the light over time, because you don't want the plants or the babies to be too shocked when they go out.
Right.
So we need to make sure that the salinity matches where they're going to, that the sediment matches where they're going to, and the light matches where they're going to. Because just moving is hard enough.
Right.
And
yeah,
so,
moving is hard.
Exactly. Yeah, so it's important for us to figure out what places in St. Thomas or in St. John need to have more mangroves, and then once we can figure out which of our babies are going to go there, then we can start to acclimate them step by step with those different conditions.
Super cool.
All right. Mason, are you ready? We're going to go check out some table stuff. Oh, he's helping, putting a shade back on,
Right.
protecting our baby plants.
Yeah. Protect the
What's happening?
babies, and then we'll go do more science. This is all. This is so much fun. I'm having so much fun. Also, I have to apologize. I realized I got a new microphone and it's super fuzzy and I'm really excited, but it keeps on like coming into the screen. So don't worry guys, I'm not being attacked by a chinchilla. This is my new microphone, which by the way, while we're moving over to the table, I want to take a quick minute and give some shout outs. First of all, I am streaming via Wi-Fi today, not from my personal cell phone network, because of an amazing grant from the Community Foundation of the Virgin Islands. You guys are amazing and huge supporters of Science Saturday. So we're really picking things up here. We're getting very fancy with my fuzzy mic and my Wi-Fi, and we're gonna have some really fun stuff coming up in months forward.
Mason, do you want to come over? The first thing that we normally do when we have these white mangroves. So this is one that's from Vesset Bay. This is our shorthand. This was the date that it was collected, so earlier this week. And what you're going to help me look at here, I'll put them out over maybe in front of you. We're going to do what's called a sprout check. And we'll see if we can, you may have good eyes and can already see some, because I'm seeing some.
So this is how I decided that I wanted to do the Science Saturday, because I drove down here one day and Kristen was sitting at the table separating seeds.
Oh, yeah.
And it seemed like the most amazing activity, and I helped, and it was so much fun. So I hope everybody is as excited about this as I am.
Okay, so basically, Allie just explained it, but we have, we're sorting these into kind of two buckets. ones that have sprouts and ones that don't. Mason, do you want to show one that has a sprout? Okay. Maybe show it into the, yeah. We can see that's the little rooting tip, and they kind of peek out. And on ones that don't have it, you can see this is it's kind of like a closed door. Sometimes they can be tricky and kind of partially open, and you have to look really carefully. But this is the first step of what we do, and we sort, which is which bucket here, folks?
Oh.
We have to be careful that we sort them into sprouted ones and unsprouted. The unsprouted ones go back into the bucket that they came from, and we'll put these sprouted ones over here. Yep. Perfect. Ooh, there's another sprout. Oh, this is one of the ones. I was saying that sometimes they can be really tricky, but that ones you can see that the door is kind of just opened.
So
You
it's
can
kinda
see the
popped
tip.
out a little bit.
Yeah, that was a good one too. Good eye, Mason. You're good at this. You're hired. You can come back and do this any day.
So one of the things when the propagules or the seeds are being collected, you want to look for signs of predation, because if the seed has been damaged, then it might not end up germinating or sprouting. And so we'll look for signs of whether there's been burrowing of different insects into the seed, or if there's signs of disease, because we don't want to bring disease back to the nursery.
Right.
Or if, like, in some places, rodents would be chewing on the seed, so looking for gnawing.
And these are some examples of what Heather was just talking about. So some of these that look like little holes, we think are like little insects that are boring into there. And that's some of the examples of one of the things that we find that's pretty common. All right.
So once we have the ones that have been separated out and have started to sprout, do those go immediately into the table to be planted?
No, we collect some data on it. So I'll finish this up, and then Allie can show you what happens next in
Okay.
terms of processing.
Okay, so I'm going to scoot these over, and then Mason, if you want to help me, we're going to start doing some processing of our sprouted propagules. So we have a data sheet here where we're taking
Data
information
sheets. I love data
on the
sheets.
propagules that we're collecting. And the reason we're doing this is because we want to know about individual propagules in our nursery. So when our trees start to grow, we want to be able to say where that propagule came from, how long it's been here, and then some other information about what the condition of the propagule was like when we first collected it. So some of the things that we're collecting data on are information about the size of the propagule, information about its color, whether or not it's floating when we got it, and its condition. So any of those things that Heather was mentioning about herbivory, or if it had nicks.
Herbivory.
Yeah, so if things are eaten.
I know you guys love when I'm talking about your science.
So, Mason, do you want to help me? We'll start inventorying some propagules. Okay. So, first we're going to write, all of our propagules get a unique propagule ID. So that's a number that tells us which number we are on in our nursery, and so we can keep all the individuals straight. So we are starting at 1951. So, right here, 51. Then we need to know, where did these come from? So we're going to work on this bucket. So, okay. So I'm going to give this a code. I'm going to say LR for the species name. These are white mangroves, Laguncularia racemosa.
Can you say that one more time?
Laguncularia racemosa.
Oh, it's written down.
It is, yeah, here. It's a mouthful.
It is.
And then Vessup, so we're going to give that a code VP for Vessup Pond. And then we're going to say the date that they were collected. 21. And then the number. Okay, so we're starting at 1951. And then these sprouted on the day that we collected them. So I'm going to write that down too, because we're interested to know, when we bring propagules into our nursery, how long does it take them to sprout? Okay, so let's get started. started on processing these propagules in this cup. So what we need to know is the weight of the propagule, its total length, and its root. So Mason, if you could help me with that, we are going to use this scale. It's a bit windy, so it might be kind of tricky.
Move around for a better view.
So normally we do our weights inside because it's a little windy, so we'll do the weights afterwards, okay? But for now, we have a sheet here so we can keep them sorted. I'll have you help me with the total length and the root length. So we're going to use this ruler. We're measuring in centimeters. So the total length is going to be all the way from that end to the very tip where you see the end of growth. So what is that?
It is 225. Can't tell really, but
Use your best estimate.
so are you including the sprout or is
You
it
are.
just the seed?
You're
All right.
including the sprout.
I'd say it's 2 centimeters. That looks like almost 3, right? Yeah.
Yep, so
2 months away.
this big mark here is the 5. So 2.5, but it's 2 marks away from that, you said? 2.7?
But
2.8. All right. And then the next part I want to know is how big just this green part is, the sprouting part. Yeah.
5 millimeters.
5. Okay. So 0.5 centimeters. Okay. Okay, and then another thing I need to know is what color is the propagule itself, so this part here, and what color is this rooting tip?
Brown.
Brown, yep.
And do you mean the berry tip or just the
Yeah,
stem?
I'm on average. So what is the color overall? Green? Brown? I think I would call this part right here.
Oh yeah, that green.
Green, yes. Okay, and then the last thing I need is how wide is the propagule? So turning it sideways and looking at it at its fattest point, what is your estimate there?
It's 1.3.
1.3, okay. Okay, and then next we want to know, does our propagule float or sink?
Ooh.
So we've got some salt water here, we're going to do a float test.
Finally, all those science experiments
It sinks.
have.
Okay. It does not float. Okay, and then if you could pull that guy out again.
So, what is the significance of it floating or not? Is that like, is it ready to do something or?
That's a good question. So
We don't know yet.
We don't actually know. So,
Okay.
we're recording all of these different metrics because we don't know a whole lot about what is kind of the best indicators for seeds here in the Virgin Islands for whether or not they're going to be successful. So we're collecting a lot of data across these different fields because we want to be able to kind of answer and figure out with our data set what's going to be the most efficient way for us to find seeds that are going to be the most successful so that we can do the best restoration work possible here in the territory.
So this is a really fun aspect of science. It's like, what else can we look at? What color is it? Does it float? All the, I can't even imagine how many more things you could ask. Although I imagine you don't want your data sheet much bigger. So for now, I think this is a really good mix.
Yeah. So the next thing that we do once we collect all that, and we missed one, we need to know, is it damaged or is it undamaged? So
It is undamaged.
undamaged.
Yep. That guy looks pretty good.
Okay. Okay, so the next thing that we do is we take a picture of our propagules that come into the nursery, and that's just so we can have kind of a photo record of what they look like when they started, and hopefully we'll be able to show you a photo record of what they look like when they grow into big trees down the road.
And today on Mangrove Glow Up, we have Propagule 1951 from Vessup.
51. Okay, so
what do we use to photo it?
I'm going to just use my phone. I'm going to take a picture on my phone. But we want to make a little tag for it so we know which one we're looking at. So can you help me with that one? We're going to write 1951. white background, so it should make it hopefully pretty easy to see the propagule. And the last thing that we want in our photo is the ruler, so that when you're looking at this picture, you can have kind of an idea of how big this propagule is. So, yep, you can set that guy on there. And then I will take the photo.
And I will video you taking the photo.
There we go. Okay, now
We did science.
make sure I
Woo!
can read the label. I can read the label nice and clearly. So the next step then is we're going to plant this propagule. So Kristen is going to show us how we do that in our lab.
Yeah. All right, hi folks. So we're gonna leave this one. This is like a cooking show, but we'll show you what it looks like over here because we have them already set up. So the next step would be to put that little propagule that we just processed. You can see these are all the little ones. They each have their tag on them, and we just push them. We make a little hole in the peat, and then we put that guy in there, and he'll grow up. Here are some other ones that are a little bit older. So these are ones that we had just planted yesterday, and you can see that they're starting to grow up into these larger plants. And actually, some of these are now ready to be transplanted. Oh, here's where we were working. So actually, Mason, you're going to be able to plant your little propagule here. So
They do.
yep. That's right. Shove that down in there pretty deep so it won't get caught by the wind. Yeah, and then that rooting tip upside down. There we go. That's actually its root. There, perfect. And that little propagule, white mangrove propagule,
You
is
did
ready to
it!
go. Way to go, Mason!
Yay!
Kind of looks like a row of almonds.
Okay.
Very cool.
So the final thing we're going to do with you all here is this next step, and Mason's going to help us with this too. So when these plants get to about this size or so, it means that they're ready to go into these bigger pots, because these things are pretty small, they're pretty tiny, they don't hold a lot of sediment or nutrients in there. So we want to give them a better place to grow. So Mason, if you want to help me, what we do is we kind of like make a little hole with our fingers. You want to do this one? And we very carefully will take these plants out, making sure we know they kind of stick together. And you're going to take this little plant, and can you plant him in here? We're going to dig down in there and take that one, just put him in. Perfect. Yeah, kind of tamp him down a little, like secure him in there. Don't be afraid. Yeah, we'll give him a little whoop, tuck him in. Perfect. And then do you want to put this label in here? Perfect. This is great. Which one do you think we should plant next? Which one do you want to try? Nice big one.
Do you want to go in order or you just pick randomly?
This one looks healthy.
Yeah.
Yeah,
that one
that,
looks really
yeah.
healthy.
Okay, so let's, do you want to take that tag out? Perfect. You can just lay it on the table while we're working. And then just very gently from the base of the plant, you're going to kind of pull it up. And hopefully it comes out kind of in a piece. Sometimes they fall apart. Perfect. Great.
A little rootsy.
Yeah, you can see them, isn't that? That's right. Isn't that so neat?
Cool.
All right. Put that one in. That's right. Yep. Nice. Perfect. You're great at this. There we go. He's ready to
Very
go.
cool.
All right.
Science Saturday meets gardening, conservation style.
Should we do one more? All right. Here, you make this hole, and then we'll, so we know that that's ready to go. Great. And then which one should we do next?
I think this one looks good.
This one?
That's a good pick.
All right. Get this And then the next step that you're going to help us with, this comes out kind of fast, is we're going to give them each a little drink. So I'll show you, this is about the one, that's about as much as we'll give them. Give them a nice start. Do you want to try to do these too?
And this is all now fresh water. So when they were floating in the table, a lot of them were in salt water.
All right.
But then once they start growing, we put them in fresh water because it's easier for them. They don't have to put in that energy trying to excrete and exclude the salt. And so that way they can grow faster. And then once they are a little bit older and can handle more stress, then we can start reintroducing salt.
Very cool.
Great job, Mason.
All right. Great
Thanks.
job, guys. All right. Well, we are coming up on just over 45 minutes. So I think, what are our final thoughts here? Mason, what do you think about this?
I think it's awesome. This is so fun. I could do this all day.
I'm hearing you guys have a new intern.
Yeah, sounds like it's me.
Yeah. All right. Awesome. So what do you guys want us to take home today? Like, what's our message?
Yeah, so mangroves, you know, they're really important. They provide a lot of benefits to people here in the U.S. Virgin Islands. We're so excited that we're building this nursery. There's good news for Mason. There's going to be lots of different ways to get involved with this nursery. So this year we're going to be building up a way for the public and the community to get involved, helping us plant these, collect the seedlings, plant them, get them outplanted into these bigger ones, and eventually outplanted into the environment here. So stay tuned. We're going to be starting to advertise for lots of opportunities to get involved, stay involved. You're already trained up. We can't wait. So thanks just so much to DPNR for having us this morning on Science Saturday. Thanks for tuning in. It was great to be here with you.
Yeah. Thanks, guys.
Thank you.
Woo! All right. Oops, sorry. All right, guys. Have a wonderful day. Have a wonderful weekend. I have no idea what we're doing next month for Science Saturday, but we'll figure it out soon. Thank you again to everyone for helping me, and to my favorite interns. You are, you know, having Aunt Kitty running Science Saturday means he has to do all of these. So, yay! All right, guys. Have a wonderful weekend. Bye.